Um, I am a graduate student in Otto Cordero's lab, and we are microbial ecologists. So we try to learn about the principles by which microbes interact with each other and their environments. And this is because microbes are the great chemical architects of our world. Through their interactions with each other, because as Erica said, microbes don't exist usually in nature as individuals, but rather as collectives, they're able to perform great biochemical feats that they wouldn't be able to perform in isolation. For example, like the degradation of biological matter into smaller usable forms that then further sustain life. Um, the principles by which microbes organize themselves into these communities are still not yet known because there are so many different complicated factors when it comes to how microbes interact with each other and modulate their behaviors. And so a major challenge still that we need to understand would be how to predict going from individuals to these collectives. And this is made difficult specifically for microbial communities because randomness reigns over life at the length scale of a microbe. Tiny fluctuations that determine how microbes are able to be available to resources or to each other can have dramatic impacts on their functions as communities. And so as humans, we often like to think about those cosmic what ifs of like, what if I did this instead of that? And then the resultant butterfly effects that could come from tiny, tiny decisions. And while we can't study parallel universes for our own lives yet, the great part about science is that experiments are ways of studying parallel universes. And so when it comes to the assembly of microbial communities, we can do experiments to study not just how communities might assemble in one configuration, but in many, many configurations by replaying the process of microbial community assembly over and over and over again. And so this is a process that we are applying specifically to studying microbial communities that are decomposers within the oceans. And so what you're seeing here is a video of marine snow shown on the left floating down throughout the water column in the ocean. And microbes that you cannot see here, of course, are populating and growing on and then degrading these particles of marine snow, which are bits of aggregated dead biological matter. And this process is hugely important, not only for liberating organic material for use by other organisms in the ocean, but also as a knob that also helps to tune climate change. And so the way that we studied the microbial degradation of marine snow in a controlled experimental replicative style would be by mimicking marine snow through only a single component of it, which is chitin, with these beads that are about 100 microns in length, which is appropriate for studying, as I mentioned, those tiny length scales at which microbial communities assemble and grow. And we inoculate these particles individually with seawater hundreds and hundreds and hundreds of times. And then we can measure several different structural and functional properties of these microbial communities. One of those things that we measured was simply the spatial arrangements of microbes as they grow on these particles by visualizing them. And what we saw when we visualized them actually surprised us. And that brings us to this image of the microbial multiverse. Each of these chitin particles were incubated and imaged separately. And so this composite image of many different particles shows you the dramatic diversity of microbial community types that emerged, starting with the same environmental selective conditions and the same initial species pools from the seawater. And just to highlight a couple particles, we can see here that there are dramatically different growth states across these particles. And this is because those random fluctuations in growth in early stages were amplified through ecological interactions between these microbes. And so there are two major messages that we can get from this image. And one of them is that randomness begets diversity. Those initial tiny fluctuations in the ways in which microbes are situated with each other and can grow with each other resulted in really, really dramatic functional differences for each of these micro ecosystems in a sense. And so this would have big impacts then on when we consider this process in nature. Which then brings us to the giant technical message, which is that the average does not represent reality. Or in other words, if we were to try to study microbial communities not at their own length scales, but rather by measuring their compositions and functions at much larger scales by, for example, considering all the particles together rather than separately, then that signal that we would measure would not represent any individual particle that we would find in reality. And so with that, I would like to thank everyone who is in the Cordero Lab, past and present, in particular the people pictured here, as well as a wonderful collaboration we're all a part of through the Simons Foundation, as well as NSF and MIT. And thank you all for your time and engagement.
Thank you.